Hello everyone. This lecture will be covering the material from Chapter 3 of your MERIB text, which covers information about cells. Okay, so let's get started. So everyone, I'm sure, has seen a cell that looks something like this in a biology class or something in high school, or biology class in college, whatever, just your like typical cell drawing. However, that's not what all the cells in our body look like. Okay, so the human body, we have over 250 different types of cells, right, that have different sizes, shapes, and subcellular components that make them unique and allow them to perform unique functions. Okay, so there's cells in our body to do everything, right? So there's cells that connect different body parts, transport gases, so like red blood cells, erythrocytes, are responsible for carrying oxygen to our tissues and removing carbon dioxide. We have cells that are able to move, so skeletal muscle, smooth muscle, cardiac muscle, all different types of muscle tissue that allows our heart to beat, digestive organs to contract, and our body just to move in general, our skeletal muscle. We have cells that store nutrients in our body, cells that can fight disease, so they can take in bacteria or pathogens and kill it. Cells that gather information and control our bodily functions, so our nervous system. Our cells are very specialized to receive sensory information so that we can perceive our environment, take that information, figure out what it means, and determine an appropriate response. And then we have cells of reproduction, right? So that's just a few, right? But there's a lot of different cells in our body. They're all going to be made up of slightly different cellular components, which allows them, again, to do their unique functions, okay? But no matter what, even though there's some individual, unique things about every cell, in all cells you're going to find the three of the same thing. So you're going to find a plasma membrane, which is what encircles the cell and is going to separate the cell from the external environment. You're going to find the cytoplasm, which is the internal fluid of the cell where the organelles live. And then you're going to find a nucleus. Okay, so the nucleus is what holds the DNA and is kind of the control center of the cell, making sure that the cell can make the proteins and things that it needs to carry out its specific functions. In some cases, as cells develop and become mature, like erythrocytes, for example, again, they become anucleated, meaning they get rid of the nucleus. So as they're developing, they have the nucleus, so they have the RNA and DNA so that they can create the proteins that they need using transcription, translation. But once they're fully mature, their function is now to carry oxygen. Okay, so the nucleus is just taking up space in that cell, essentially. So the cell gets rid of the nucleus so that it can carry more oxygen, because once it's fully developed, it doesn't necessarily need the nucleus. Okay, but in all cells, you're going to find those three main components. And then obviously, depending on the cell type, there's going to be different stuff. So this is just your general, cell that you would see again in a book. So again, you're always going to have a nucleus in a cell, a plasma membrane in a cell, as well as cytoplasm. And then all this other stuff, most cells you'll find the majority of these things, like mitochondria, lysosomes, the endoplasmic reticulum, ribosomes, so on and so forth. But you'll just find different amounts of these things in different cells, and also some more specialty organelles that are important in that cell's particular function. So we're not going to worry too much about all the functions of these different organelles, just the general ones. If you're in just like a intro bio class, you're going to spend more time on that. But for our purposes, we're going to just leave it at this. Know that there's different organelles found in different cells. You should know like the basic organelles, I would think. But when we get into the more specific cell types, then we'll go into more detail about what organelles are really involved in the actual physiological happenings in the cell. So Don't worry about all that for right now. Okay, so outside of the cell, we have our extracellular matrix. And the extracellular matrix is everything that acts as the glue that holds the cells kind of together, right? So obviously we have individual cells in our body, but outside of the cells, there's also a number of different proteins and molecules that kind of hold everything together and fill in the empty space, okay? So there's extracellular materials, right? The matrix is kind of the whole thing collectively, but we also have extracellular fluids that make up the extracellular materials. Extracellular fluids are just bodily fluids. So some example, interstitial fluid is all the fluid that's surrounding the cells. So cells need nutrients, they need oxygen, they need sugars for energy. The interstitial fluid is a fluid that is all over our body and is responsible for kind of bringing those different nutrients, sugars, whatever it may be, to the cells. Okay, so the cells are going to be always in contact with some sort of fluid, and that allows them to absorb what they need when they need it. Another fluid that we have is our blood plasma, and that's just the fluid of our blood. So our blood cells, again, are floating in that specific kind of extracellular fluid, the plasma. And then lastly, the cerebrospinal fluid, which would be the fluid that surrounds our nervous system organs. Okay, so our brain and spinal cord, we have ventricles or the central canal running through those structures. And in the central canal and ventricles, we have CSF, or cerebrospinal fluid. And it's kind of like the same function that I talked about for the interstitial fluid. It brings nutrients, sugars, things to the cells of the nervous system that they need to function. Okay, so really important to make sure that the cells get the nutrients and everything that they need. And that fluid also acts kind of like a cushion and helps cushion the brain from trauma. Also, outside the cell, we can find extracellular secretions. So some of our cells are able to secrete substances. We have digestive substances, saliva, things that are important in digestion of food. And we also have some lubricants in our body, mucus and whatnot through our digestive organs and other parts of our body that make sure things passing through can go smoothly. Okay? So that's what's outside the cell. And again, like I mentioned earlier, the plasma membrane or cell membrane is what's responsible for separating the inside, the intracellular fluid, from the outside, the extracellular fluid. Okay, and it's really important for cells to have this membrane so they can control their internal environment. Okay, so the plasma membrane is really cool. It plays a dynamic role, meaning that it's changing constantly, and it controls what enters and what leaves the cell. Okay, so even though a cell is bathed in that interstitial fluid, say there's a lot of sugars in that fluid, the cell doesn't want too many sugars if it doesn't need them, so that plasma membrane controls what comes in. It's only going to allow stuff in that it needs or that it wants for some purpose, right? Our plasma membrane is made up of lipids, those phospholipids that we talked about in chapter 2, that make a lipid bilayer. Okay, so those lipids are lined up. I'll show it in a few slides, but they form sort of like a fluid layer, right? So the lipids can move around and stuff can pass through, but it's semi-permeable, which means that only certain things are allowed through. Okay. Inside of our plasma membrane, we also are going to see a number of different membrane proteins, which again we'll talk about in a minute here. But these membrane proteins are going to be floating through the membrane and doing different functions. So we'll talk about all the different functions they do. But because it can kind of float through that lipid bilayer, the membrane itself, the pattern is constantly changing, right? So you're not going to see a cell and it's always going to look exactly the same. The shape of it's going to change a little bit. Where the proteins are located is going to change a little bit. And that's why it's referred to as the fluid mosaic model. Okay, and another really important thing that, again, all of these things we'll talk about in more detail in a minute, but something that's also very important for our cell membrane is these structures called cell junctions that are part of the cell membrane and made up normally of these proteins. But the cell junctions are going to help hold our cells together so that we can actually have solid organs and tissues and whatnot in our body. Okay, so here's an image of what your cell membrane kind of looks like. So I'm just going to zoom in. It's a little blurry, but so you have your phospholipid bilayer, right? So you have the phospholipids have the phosphate head and the fatty acid tails, and it's a bilayer because there's two phospholipids in the layer, right? There's two layers, two bilayer. Okay. So that's the phospholipid bilayer. And then some other stuff you can find in here. So we talked about proteins a little bit already. So all these purple guys are considered proteins, right? And they're embedded in this fluid membrane, okay? So they can move around, do different things. And we'll talk about all the different kinds of proteins with their different functions in a minute. We also have this cholesterol embedded. So you can kind of see these little yellow boys. Those are all cholesterol molecules. Those help stabilize the membrane, right? So I said before it's fluid. So those cholesterol molecules allow the membrane to become a little bit more solid, so it's not quite so fluid, okay, which is important. And then finally, carbohydrates. So sugars, these green things, right? Carbohydrate is just a polysaccharide or complex sugar, right? So you're going to find a bunch of carbohydrates on the outside of the cell surface, and we'll explain why that's important in a second as well. Okay, so that's the basic makeup. So now let's get a little bit into more detail about all these different parts. Okay, so I mentioned our phospholipid bilayer is made up mostly of those phospholipids that have the phosphate head, right, and the fatty acid tails. We talked about this in chapter 2, but the phosphate heads are polar, okay, which means they're charged. The tails are nonpolar because they're the fatty acids, okay, and if you remember, water is also polar, right, because water, we had a polar covalent bond, unequal sharing of the electrons, right? So the polar head of the phospholipid, because it's polar and water's polar, we refer to this as being hydrophilic, which means it loves water, okay? Because it's polar, water's polar, it's going to be attracted to water, okay? The opposite, these tails, since they are nonpolar, they don't like water, we call those hydrophobic. Okay, water fearing. Okay, because they're nonpolar, water's polar, those don't get along. Okay, so the majority of the bilayer is made up of those phospholipids, and because of their polar heads and nonpolar tails, they line up in a way where the nonpolar tails meet each other on the inside of the membrane, and the outside are all the polar head groups. Okay, so you have the polar heads exposed to the extracellular space and the polar heads exposed to the intracellular space, right? Because these spaces are all fluid, right? So they're mostly water with some other things dissolved in them. So the polar heads are attracted to the water on the outside and inside of the cell. Okay, so it allows the membrane to form this barrier between the liquid outside the cell and the liquid inside of the cell. We also have some glycolipids, which are lipids that have sugars on the outside, and then cholesterol, which, like I mentioned previously, just increases the membrane's stability, makes it a little bit more rigid. Okay. So that's the cell membrane. In the membrane, we also have a number of different proteins that are going to be embedded. The proteins have a bunch of different functions, which we'll go over in a minute. But basically, their function is to allow communication with the environment. So figure out what's outside, kind of sense the environment, allow stuff to move in or out of the cells, communicate with other cells, hold cells together. There's a lot of different functions. Proteins are really big, and they actually make up about half of the mass of the plasma membrane. And like I said, they all have different functions. There's two main types of proteins, integral proteins and peripheral proteins. So I'll show you what those are in a second. Integral and peripheral. So integral proteins are going to be proteins that are firmly inserted into the membrane. Okay, we often refer to them as transmembrane proteins, which means they span the length of the membrane. So if, I'm not going to draw, I'm just going to go back and show you these guys. So an example of a transmembrane protein, these long boys that go the whole length of the membrane would be considered transmembrane proteins or integral proteins. Okay, so integral or transmembrane proteins. To be embedded in the bilayer like that, they have to have a hydrophobic and a hydrophilic region. Okay, the phobic region is going to interact with the tails of the lipids. Hydrophilic obviously is going to interact with the water and the phosphate heads of the phospholipids. Okay, and they have a bunch of different functions. I'll tell you those on the next slide in more detail. The other type of protein we have are peripheral proteins. Instead of being inserted into the membrane, these are loosely attached to the other proteins and kind of sit on the surface. Okay, so I'm going to go back really quick again and show you. So the peripheral proteins are these guys that are going to be just on the surface, right? They're attached near the surface. Okay, so the peripheral proteins a lot of times have these filaments used for support. And again, a lot of different functions, and we'll talk about those functions next. But integral are through the membrane. Peripheral are just attached kind of on the outside of the membrane. Okay, so this is blurry, but some of the functions of the proteins, one which we'll talk about in more detail in literally a few slides here, is transport. So we have different channels or carriers that transport materials in or out of the cell that are too big to pass on their own. We have receptors that the cell communicates with other cells a lot of the times through chemicals or ligands. And the receptors can attach to those ligands, and it can cause some sort of response. You can have proteins that have enzymatic activity, cell-to-cell recognition with the glycoproteins. We have just proteins that are places for attachment to kind of help stabilize the cell. And then proteins that are functioning in cell-to-cell joining, so connecting cells together to help stabilize those. Okay, and a lot of this we're going to talk about in more detail here, so I just skimmed through this, but I would definitely look through this in your book. The book gives good examples of all of these. Okay, so I mentioned here cell-to-cell recognition. We have these glycoproteins that help the cell become recognized. Okay, those glycoproteins, along with glycolipids, are what's responsible for forming what we call the glycolics, okay, which is just a fancy, I guess you could call it maybe like the fingerprint of the cell. It helps the cell become recognized. Okay, so glycolipids and glycoproteins are just either lipids or proteins that have a sugar or carbohydrate sticking out on the surface. Okay, so I'm going to go back again. I'm sorry, I keep going back. But these, right, the carbohydrates on the surface, so this would be a glycoprotein because you have a protein attached to the carbohydrate at the surface. Okay, and those carbohydrates or sugars sticking out of the surface make the cell unique. Okay, it's a unique sugar coating that's individual for each cell. Okay, so it allows cells to recognize each other most importantly for our immune system, it allows our immune system to know what's our own cells and what's a intruder, pathogen, bacterial, whatever cell, okay? So the immune system is responsible for patrolling our bodies, finding any things that shouldn't be there and getting rid of them, so keeping us healthy, right? And we don't want the immune system to attack cells that belong in our body, okay? Because that would not be good, right? That would be an autoimmune disorder. So this glycolyx allows the immune system to figure out what is our own cells that are supposed to be there and what are the cells of the invader that actually need to be attacked. So the glycolyx is really important. Again, glycolipids or glycoproteins is what makes up that sugar coating. Okay, another function of the proteins that we talked about is forming cell junctions, which are just connections between cells. Okay, so in our body we have some cells that are free. So think of like your blood cells, right? Your red blood cells are just floating around in your plasma. So they're not connected, shouldn't be connected to other cells unless there's a clot or something. Sperm cells are another example of that. But most cells are bound together. So think we have tissues and organs in our body that are like a solid mass. So there has to be some sort of connections between these cells to make this solid mass of tissue and to make these organs that can function, like your heart, to be able to pump as one organ. All these cells need to be able to communicate and be held together. Okay, there's 3 different types of cell junctions that we're going to find. Okay, we have tight junction, desmosomes, and gap junctions. And they all have different functions, but essentially it's allowing cells to be joined, either just to keep them stable or for communication. Okay, so let's go through those quick. So the first one is our tight junctions, and the name kind of tells you what it is. So it is forming an impermeable junction between cells. So it's kind of hard to see here, but these little purple guys here would be where you're forming your tight junctions, and it's hard to see, but it's going all the way around the cell. So this cell would be connected to the cell to its right, to its left, the cell behind it, and the cell in front of it, if you will. And it's forming these really tight junctions that are impermeable. Okay, and this is really important, especially in certain cells, because it prevents fluids and molecules from moving in between cells. So you're going to see this especially in cells like your epithelial cells lining your digestive tract. Okay, so say up here, this is your digestive tract. These are the epithelial cells lining it, right? So you have food passing through your digestive tract here. To digest food, you need a number of things. You need enzymes to break down the proteins, to break down your starches and the fats. And then when you're eating food, you're taking in material from the outside, right? So there's potential that this food is dirty, or there's microorganisms or bacteria, whatever it may be. So these tight junctions are really important in areas like this, because we don't want either digestive enzymes or bacteria, whatever it is, from our GI tract going through these cells and entering the bloodstream, right? Because below this, you're probably going to have some sort of vascular system. So if the bacteria or whatever can get through between these cells, it's going to enter the blood and get to the rest of our body. So these tight junctions prevent that from happening. So they're really important for that. Okay. The next type of cell junction we have are called desmosomes, and they are a rivet-like cell junction. They're these specialized proteins called cahedrons. They're going to lock into the neighboring cahedrons and form like a zipper. Okay, so if we look at, they're between here and here, but if we zoom in on this, So you'll see this cell has these cohedron proteins. This one has these cohedron proteins, and they're going to kind of overlap and form this zipper that's going to kind of Velcro those two cells together. Okay, so it's really important to connect to the cells. You see these proteins are anchored to a plaque on either side in the cell. Okay, desmosomes are an important connection because they allow some give between cells, reducing the possibility of tearing under tension. Okay, so they're similar to tight junctions in that they hold the cells together, but we don't want the junctions to be too tight in areas that we have a lot of stress. So areas like our skin or our cardiac muscle, your heart muscle, right? There's going to be a lot of cells kind of moving around and a lot of mechanical stress, right? You can pull on your skin, your heart muscle pumps, fills with blood, pumps the blood out. So there's a lot of movement and stress on those cells. And we don't want just tight junctions that can't move at all. So these desmosomes still allow the cells to stay together, but allow them to kind of move and get pushed around without just tearing apart. Okay, because the tight junctions, if you put too much stress on the two cells that are held together by tight junctions, this will just tear. Okay, there's no give at all because we want that to be impermeable. The desmosomes allow some movement. Okay, and then the very last type of cell junction are called gap junctions. These are also transmembrane proteins, but they form tunnels between cells that allow small molecules to pass between cells. So things like ions, sugars, and other small molecules need to move from cell to cell, especially in cells that use a lot of electrical signaling. Okay, so for example, it says right here, cardiac and smooth muscle, you're going to find a lot of desmosomes because in those cells, the muscle cells, you want all of the muscle cells of your heart to contract at the same time, right, to pump blood efficiently. So these desmosomes, these channels, allow that to happen. So you see that you have the integral proteins on each plasma membrane, and together they form a channel so things can move between cells. So the ions we need for contraction to happen of our cardiac muscle, these gap junctions allow those ions to be passed rapidly so that communication can happen quickly. Okay, so those are our three junctions, our cellular junctions, which concludes the portion about the plasma membrane. Okay, our plasma membrane, we have obviously a semi-permeable membrane, which I mentioned before, that allows only certain materials to pass through. Okay, our cells need a bunch of different stuff to come in or leave the cell in order for the cell to function properly. So we need to be able to get substances across the cell membrane. Okay, so certain molecules can just go through on their own. Certain molecules are going to need some sort of transportation mechanism to get through the membrane. And there's two different ways that substances are going to cross the membranes. So we have passive transport that requires no energy. And active transport, think you're being active, you need energy. Okay. Passive transport, it's on the next slide. Passive transport, like I said, requires no energy input. Okay, so passive transport is going to rely on diffusion for molecules to be moving. Okay. Hopefully everyone's heard of diffusion before, but diffusion is just the natural movement of molecules from an area of high concentration to an area of low concentration. Okay, so even if you've not really heard too much about diffusion, I'm sure you've experienced it. If you have an air freshener in your house, the smell from that air freshener is going to be highly concentrated right where the air freshener is plugged in, right? And that smell is going to diffuse through the air and go from the highly concentrated plug-in zone to a lower concentration, say in a different part of your house, right? But that smell is going to move because it's diffusing from high concentration to low concentration. Okay, we refer to this as moving down a concentration gradient. Okay, and this is going to be the basis for our passive transport. Passive transport is going to be the movement of molecules in or out of the cell using diffusion. So using just the concentration gradient of those molecules to drive the movement. Okay, and there's three different types of passive transport. We have simple diffusion, facilitated diffusion, and osmosis. Okay, so all three, again, passive, you need no energy. It's just the concentration gradient and the desire for the molecules to want to spread out that allows the molecules to move in or out of the cell. Okay. So another example of diffusion, so I gave the air freshener example, but if you were to put a tab of dye in a beaker, or say like a drop of food coloring, we have an area of high concentration right where that dye is, eventually the dye is going to spread out, right? So in this one, we have evenly spread out dye. When we start, all the dye is going to be around where we put it, and it's slowly going to diffuse away and move from areas of high concentration at the tablet to areas of lower concentration. Okay, and that's diffusion. And that's what's going to happen in any sort of passive transport mechanism in our cells. Okay, we're just using diffusion to move substances in or out of the cells down their concentration gradient. Okay. The speed of diffusion can be influenced by three things. The first is the concentration gradient itself. Okay, so the greater the difference in concentration between two areas, the faster the diffusion occurs. Okay, so in this example, if we put a very strongly concentrated dye in the bottom, it would diffuse more quickly than just a little drop of a diluted dye. Right? Because there's more molecules of dye in that area. They have a higher concentration, so they want to move apart more quickly. The second thing that influences the speed is the molecular size. Smaller molecules diffuse more quickly. And I always give the example, which has nothing to do with diffusion, but I imagine little puppy and Shrek in a mosh pit at a concert or something. And they want to get out of the mosh pit. They're both in the middle, want to get out. The puppy is small, can easily weave through the legs of the people, go around people, sneak out, where Shrek is really big and has to say, oh, excuse me, that's what I imagine he would say, to get out of the mosh pit, right? So the smaller puppy can go more quickly. The big old Shrek takes longer to get through. Okay, so smaller molecules diffuse faster. And then finally, the last thing that can affect the rate of diffusion is the temperature. So as we increase temperature, we're increasing the kinetic energy. And the kinetic energy is just the random movement of all molecules at all times, right? So if you have a glass of water, in that glass of water, there's little molecules and they're all moving around. Okay, so that's just kinetic energy. It happens naturally. That's what's leading to diffusion, is just moving around, the kinetic energy. As we increase the temperature, we're increasing kinetic energy, which is going to result in faster diffusion. Okay. When there is no net movement of molecules in one direction, we have reached equilibrium. Okay, so there's no longer a concentration gradient if there's no net movement. Okay, so again, if you're moving from an area of high concentration to low concentration, obviously molecules are moving. Once you've reached equilibrium and everything's fully diffused and there's no areas that have more high concentration, then we've reached equilibrium. Okay? So, what can diffuse? Okay, so this is all types of, again, passive transport. We have simple diffusion, which is lipid-soluble and non-polar substances, very small molecules that can easily pass through the membrane. So simple diffusion, you're going to have molecules going directly through the membrane, and things that are lipid-soluble are able to get through the tails of the phospholipids, okay? And other very small things. Even if they're polar, they might be able to get through. Facilitated diffusion is for larger or non-lipid soluble, which is polar, molecules, okay? So those things can't just go through the membrane on their own, right? Because if they're polar, they can't get through that lipid tail hydrophobic region because that's non-polar, doesn't mix very well. So they need some sort of carrier molecule. So some sort of protein carrier or channel that allows them through. And then the last type of passive transport is osmosis, which is sort of kind of a type of diffusion, except instead of the solute moving, the solvent, which is normally water, is moving. Remember, in our solution, we have a solute and a solvent. Solvent is normally water. A solute is anything that is going to dissolve in the water. So some sort of salt, a sugar, things that are just dissolving are the solute. The solvent is the water. So when we talk about diffusion, we're talking about the movement of the solute. Osmosis is the movement of the solvent. Okay? Both of them, though, are not going to require energy. They're all going to be a type of passive transport. Okay, so let's talk a little bit more about these three types of passive transport. So simple diffusion, like I said, you need non-polar, lipid-soluble, hydrophobic molecules to do simple diffusion. So again, remember in the middle of that lipid bilayer, those tails are going to be non-polar, hydrophobic lipids, right? So in order for something to get through that middle part, hydrophobic area, it also has to be polar, okay? So only non-polar molecules can go through simple diffusion. So simple diffusion is just the molecules are going directly through the bilayer. Okay? So again, non-polar, lipid-soluble, hydrophobic. They all mean the same thing. So those small molecules can go directly through. So oxygen, carbon dioxide, some hormones, and fatty acids. However, there are cases of small polar molecules, such as water, that can pass through passively, okay, without any sort of carrier molecule. So even though polar molecules typically wouldn't be able to get through, again, the issue is this middle part where it's the fats lined up. Normally polar molecules, once they try to go through the polar heads, once they hit that hydrophobic area, they'd be booted back out. But if they're small enough, they might be able to get through. Okay, so that's simple diffusion, just a molecule going directly through the lipid bilayer. Facilitated diffusion is for hydrophobic molecules that are very large, that go passively down their concentration gradient. Okay, or polar molecules as well. So we have carrier-mediated facilitated diffusion and channel-mediated facilitated diffusion. Okay, so I'll tell you what each of those are. So carrier-mediated, you have a carrier that is a transmembrane integral protein. So in this image, remember a transmembrane, I mean it goes through the whole membrane. Each carrier molecule, again, it's just a protein, is going to be able to bind to a specific solute of some sort. So in this case, say this is some sort of sugar. The sugar is too big to get through the lipid bilayer on its own. So instead, we have a specific receptor here, a specific carrier that the sugar is going to bind to. Once the sugar is bound, the carrier is going to have a conformational change of shape, right? So it's going to change shape and transport that sugar somewhere else, okay? And again, each carrier only works with a certain type of solute, okay? It's not going to work for everything. Only certain things can get through, okay? Obviously, the number of molecules that can get through is dependent on the number of carriers present, okay, because they can only transport one molecule at a time. Once they're fully saturated, that means they're bound to as many molecules as possible, busy transporting, okay? So at some point, there's going to be a limit on how many molecules can be carried through, okay, because it can only do one at a time, okay? And again, remember this, we're still talking about diffusion here. So the movement of these sugars is based on its concentration gradient. So you see there's 5 sugars outside, only 2 inside, on that side at least, right? So the sugars are going to move down their concentration gradient from an area of high concentration to an area of low concentration. Okay, so no energy is required. We also have another type of facilitated diffusion called channel-mediated. And channel-mediated, instead of being a carrier, now we just have channels, okay? Channels, again, are integral proteins, so they go through the lipid bilayer, but they have a channel that's just open in the middle of it that ions can flow through. Okay, and they do this by, in the middle of their channel, you have an aqueous filled core, which means it's polar, so polar substances can move through it easily. Okay, the channels transport molecules such as ions or water, again, down their concentration gradient. It's really important you remember that all of these are examples of diffusion, so it's going to be down a concentration gradient. So in this case, you have a high concentration outside of the cell of whatever the solute is, a low concentration inside the cell. So the solutes can move from an area of high concentration to an area of low concentration. Okay? These channels are specific for what it allows to go through based on its pore size and the charge. Okay, so if we're transporting ions, remember things like potassium with a positive charge is a cation. Chloride has a negative charge, it's an anion. Some channels only allow certain things in or out, okay, depending on the size of the ion, the charge of the ion, or whatever, okay? So it's going to have a specific substance that it's allowing. Okay, and there's 2 types of these channels, which we're just going to skim over right now, but we're going to get really into this during when we talk about muscles and the nervous system. We have leakage channels that are leaky, so they're just always open. Okay, so this one here in the image would be probably considered a leaky channel. So whatever the solute is, it can just move whenever the concentration gradient changes. Gated channels are still channels, so they have that pore in the middle that ions can go through, but they're required to have a chemical or electrical signal in order to open. So we can have ligand gated, which means chemical, so things like neurotransmitters, or voltage gated, which again we'll talk about in the nervous system and the muscle physiology, but when we talk about action potentials and depolarization of the membrane, we have voltage gated channels that are going to respond to different changes of voltage. Okay. The last type of passive transport is osmosis, which is similar to diffusion, but instead of the solute moving, the solvent is moving. Okay, so most cases that's going to be water. And it's moving across the selectively permeable membrane. So sometimes it can get through just through simple diffusion on its own. A lot of times it's going to move through a certain channel called an aquaporin, which is just a channel specifically designed to allow water to move through. So I got ahead of myself, but it can move through the lipid bilayer on its own or through the aquaporin. The flow of water occurs when the concentration of water is different on two sides of the membrane. Okay, so again, osmosis is just the movement of water. So now we're looking at the concentration of water, but we can also speak about it in terms of the concentration of solute. Okay, so when we talked about molarity briefly in chapter 2, we talked about it a lot more in lab. Osmolarity is a way to measure concentration of solute. Okay, so when we look at say a red blood cell, I don't know if Zach went through this with you. Red blood cell has an osmolarity of 300 milliosmol. Okay, that's because there's different numbers of ions, different sugars and salts and stuff in the blood cell that's going to give it this concentration. Okay, say we have that blood cell going through our bloodstream and our plasma, our blood plasma. Say we have a 200 milliosmol concentration of the plasma. Okay, so there's a difference in the concentration. Again, the osmolarity is looking at the concentration of solute. Water is going to want to move from an area of low solute concentration to high solute concentration. Okay, so for this example I just drew, the plasma has a low solute concentration. The blood has a high solute concentration. So water in this case is going to want to move into the cell. Okay, so I like to think of it like the water is going to dilute these salts and stuff in the cell, right? Because we have a higher concentration of salt in the cell compared to out of the cell. So water is always going to move from areas of low solute concentration to high solute concentration. Okay, we can think of this in, let's say, I do the same thing, but instead now we're doing we have inside the cell, say it's 60% salt. This is not scientifically accurate. Outside the cell we have 20% salt. Okay. So with that, you should be able to determine, just like the first example, same thing. If we have a higher salt solution, higher concentration salt solution inside the cell, water is going to want to go into the cell to dilute that. Okay, because there's less salt outside, more salt inside. It wants to dilute that salt. We can think about it reversely too. We have 60% salt, so we have 40% water in the cell. 20% salt outside is 80% water, right, to get 100% total. Okay, so it's moving from low solute to high solute, or vice versa, moving from a high water to low water. Okay, so we have a lot of water outside, not much water inside, so water is going to want to go inside. Okay, so there's two ways to think about that. Whatever makes most sense in your head. I know people can get confused easily, but when you're doing a problem or something, just make sure you're paying attention to the solute. Are you talking about the solute, or are you talking about water or the solvent? Okay, because they're two very different things. All right, so if you have a semi-permeable membrane here, here's an example of kind of a setup. We have a membrane. See this guy here. We have a setup. We have a membrane that allows anything to pass through. And our initial starting concentrations, we have a lot of solute sugar molecules on one side, not many on the other side. Right, so you're going to have diffusion happening. So the sugar is going to move from areas of high concentration to low concentration. Okay, so what you're going to see is this sugar diffuse to areas of low concentration to create an equilibrium. Let's put equal. Okay, equilibrium. Okay, so that's just what diffusion is. With osmosis, a lot of the times in cells, you're going to see that we have an impermeable membrane to solutes. So now, the only thing that can move through this membrane is water, or the solvent, right? So we have a high concentration of the sugar, again, on this side, a low concentration on this side, but now the sugar is not able to move. So instead of the sugar moving, we still want to create an equilibrium, regardless of if sugar can move or not. Because sugar can't move, water is going to move instead. Water is going to go from an area of low solute concentration to high solute concentration, again, to create an equilibrium. So there's equal parts sugar to water, okay? And then this side, we are at equilibrium. Okay? The movement of water, don't stress out too much about this, but there's two different pressures that play a force on the movement of water. So hydrostatic pressure is an outward pressure exerted on the cell side of the membrane, caused by increased volume of the cell due to osmosis. So if we have our cell that has a bunch of water in it, right, the water is going to push onto the outside of the cell, right? Especially if we get more and more water, the hydrostatic pressure is going to increase because the water wants to leave the cell because the cell can only hold so much. Osmotic pressure is the inward pressure due to the tendency of water to want to be pulled into the cell with higher osmolarity. Okay, so at rest, there's water in the cell that kind of wants to get out. But also in the cell, we talked about there's a bunch of different ions, salt, sugar, whatever stuff in the cell that makes the cell have some sort of internal concentration. Osmotic pressure, I'll use purple to show. Osmotic pressure is the want for water to go into the cell to dilute those solute particles. Okay, so hydrostatic pressure is water wanting to get out. Osmotic pressure is water wanting to get in. When you get to 208, you'll talk more about these forces, especially when we talk about the blood levels, blood pressure, and the urinary system too. Okay. And then the last bit here, talking about osmosis and the movement of water. This term, tonicity, is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. Okay, so if we're in a situation where water is wanting to enter the cell or leave the cell, we can call the solutions, they have different tonicities. Okay, so first the definitions, then I'll show you drawings. An isotonic solution, you have the same osmolarity inside of the cell as the solution. Okay, so all these is talking about the solution. Okay, so you're already at equilibrium, so there's not going to be a change. You can have a hypertonic solution, where the solution has a higher osmolarity than inside. This results in water flowing out of the cell. If water is leaving, that's going to cause the cell to shrink. Shrinking is known as crenation. And then the last type of tonicity you can have is a hypotonic solution. Hypotonic, the solution has lower osmolarity than inside the cell. So water goes into the cell. If water is entering the cell is going to swell. If too much water enters, the cell can burst, and that's called lysis. Okay, so I would start by understanding those definitions first and foremost. Okay, but I think drawing all this out helps understand it a lot as well. So what I'm going to do is draw blood cells in 3 different solutions. So we have, say we have blood cells. I'm putting them just in a beaker. This is terrible, but you get the point. Okay, the internal concentration of our blood cells is 300 milliosmol. Okay, so that's not going to change. But we can have solutions surrounding the blood that either has a greater concentration, a lower concentration, or the same concentration, right? So in an isotonic solution, our concentrations are the same. So outside of the cell, the solution would have a 300 milliosmol concentration as well. Okay? Again, remember in all of these situations, we have a semi-permeable membrane which only allows water to move. Okay, so water can move in and out of the cell, right? Not a problem. But if we have equal concentrations, there's not going to be any net movement of water, meaning the cell's not going to gain water, it's not going to lose water, it's just going to stay the same because we're already at equilibrium. Okay, so that's an isotonic solution. Hypertonic solution, you're going to have a solution that has a greater concentration than inside the cell. Okay, so when we talked about osmosis, water is going to want to move from an area of low solute concentration. I'll do this in a different, that's the same color I used, Claire, to a low solute concentration to a high solute concentration, right? We have more ions outside than inside, so water is going to want to move out to dilute that. So this is considered a hypertonic solution. The cell is going to lose water. The last example is the opposite of that. So if we have a solution that has a concentration lower than the cell, again, we're not at equilibrium, so water is going to want to move. Water is going from a low solute to a high solute concentration. So water is going to go into the cell and try to dilute that internal concentration. Okay, in a hypertonic solution, well, in isotonic, nothing's really going to happen because it's already at equilibrium. Hypertonic solution, the cell is going to shrink, and we call that crenation. In a hypotonic solution, the cell is going to swell, and if it gains too much water, eventually that cell will burst, and we call that lysis, when the cell bursts. Okay, and I remember that hypotonic is this setup because I think the water is entering the cell's getting big and fat. I think hippo, the cell's getting big and fat like a hippo, it's swelling up real big. So hypotonic, I think a hippo. Okay, so those are the 3 tonicities and what would happen if you had a cell in each of those types of solutions. And this is just the same thing, but with images of red blood cells, okay? It's actual ones, not my drawing. So again, isotonic solution, we're already at equilibrium, so there's not going to be a net movement of water, so your blood cell is going to stay just the same. Hypertonic solution, we have a greater concentration of solute outside compared to inside, lower concentration inside. So water is going to leave to try to dilute that external solution. So the cell is going to crenate or shrivel up, and this is what a crenated red blood cell looks like. Hypotonics, the opposite. We have a higher concentration of solute inside, a lower concentration of solute outside. Water is going to go into the cell to dilute that higher concentration. So the cell gains water, gets big and fat, think hippo, right? And eventually, if that cell gets big enough, it could burst, and that's called lysis. Okay, so those are the tonicities. All right, I'm going to stop here for the first part of Chapter 3. There will be a second part of Chapter 3 covering active membrane transport.